Good afternoon, everyone. And for the people online, can they say something in chat if they can hear me? We would like to begin this seminar as normal with our land acknowledgement. Iowa State University aspires to be the best land-grant university at creating a welcoming and inclusive environment where diverse individuals can succeed and thrive. As a land-grant institution, we are committed to the caretaking of this land and would like to begin this event by acknowledging those who have previously taken care of the land on which we gather. Before the site became Iowa State University, it was the ancestral lands and territory of the Bakoje or Iowa Nation. The United States obtained this land from the Meskwaki and Sauk nations in the Treaty of 1842. We wish to recognize our obligations to this land and to the people who took care of it, as well as to the 17,000 Native people who live in Iowa today. Now I would like to introduce Dr. Reuel. Dr. Nigel Reuel is an associate professor of chemical and biological engineering at Iowa State University. He received his PhD in chemical engineering at MIT in 2014 under the guidance of Michael Strano. After graduating, he attempted to commercialize his PhD work in a startup, Soapbox Biologic Incorporated, and then consulted at a larger life science tool company that obtained the startup's intellectual property. He then worked as a research investigator at DuPont's Central Research and Development Campus for 2 years. He was then promoted to the Corporate Technology Scout, where he traveled to universities and incubators to find technology for DuPont. He quickly realized that making technology is more stimulating than finding it, and was pleased with the opportunity to come to ISU and become an entrepreneurial-minded professor. Currently, his group has over 15 active technology disclosures at ISU RF, 2 granted ISU patents, and 3 startup company offshoots. Dr. Reuel's work has been recognized by the NSF Career Award, NIH Outstanding Early Investigator Award, 3M Non-Tenured Faculty Award, and the BMES Advanced Biomanufacturing Junior Investigator Award. With that, please join me in welcoming Dr. Reuel.
Thank you for that intro and for recognizing the land we stand on. I appreciate that. Thank you for this invitation. It gave me an opportunity to put together some slides that are new, so they may be a little bit rough around the edges, but this is not typically the papers I get to highlight. So this is exciting that I get to do that today. Yeah, so as my title shows, I am probably the odd person here. So I'm a chemical engineer standing amongst many soil scientists. So I thought I'd just give you a perspective that comes from mine. And actually the first time I came to Iowa was as a technology scout. So many times to Johnston, Iowa, working with Pioneer, now Corteva. Also on a similar subject of how do you look beneath the canopy? So I'm glad to continue this conversation. All right, so in terms, oh, hang on, I'm on Zoom, I gotta click that here. All right, so as a chemical engineer, you might think of us more like this, right? Where you have a big chemical plant. So you go down to Eddyville and, we send students here and we're on the back end of what comes off the soil. We train our students classically to think a lot about pumps and different unit operations. And we draw these process flow diagrams of how do you go from inputs to outputs. And this is a very important agricultural one. So it's the Haber-Bosch process responsible for creating much of the fertilizer we use. So lots of chemies are classically here, but I'm a bit of a renegade. I think of different chemical processes. So instead of really big plants, I think of little plants. So treating living organisms same way as a factory. So this little diagram shows a cell. So, inputs coming in, outputs coming out. But really what I'm here to talk about today is more than just a single cell. It's the complex system that we call soil, right? That has so many different things. And as I've gotten to work with Marshall, have become more and more entrenched in this area of how important soil is, as well as how complex it is. And so it's really fun to come in as a chemical engineer to think about this very basic premise we have of a worldview of inputs going to outputs, and how you can measure those. So with a chemical plant, there are sensors all over the place. So I teach, one of the courses I teach is process control. And you can't do any process control unless there's some way to measure things. And it's similar in terms of field management, how the better we can measure, especially in this very heterogeneous environment, the better off we can come up with novel and new control strategies and implement those in the field. And that comes with the caveat very significantly here with agriculture is the cost constraint. So I first came aware of this as I was working with Corteva before DuPont Pioneer. Just the vastness of a field and how diverse it is, and if you wanted to put sensors all over, kind of just how low cost that needed to be. And so a lot of work that we do in my group kind of carries that theme with it of practicality of, okay, it's great you can measure something, but how could you translate this to make thousands of measurements? How could you actually implement this? And so hopefully I can highlight some of that work that we've done. Of course, I need to acknowledge those who have done this great work. So I've got a team of very talented students who are currently working with me, as well as those who have gone on. Some of the work I'm going to specifically highlight today is work of Dr. Charkabi, who's now at 3M, Dr. Kalmeyer, who founded a startup, one of those that are an offshoot of our group, so he's here still in Ames, and then a little bit work of Adam Carr, who will be leaving our group. He had a very interesting opportunity to take some of our sensors and work with Corteva over last summer and implement them and put them out in the field. And those were our deep soil moisture monitoring sensors. So I'll highlight some of that work. And of course, need to acknowledge funding. Can't do this without paying the bills. So very grateful for all our funding agencies. Okay, so one of the things that ties our kind of crazy group together is thinking about closed systems. So if you think about sensing, if you break it down, really, you're trying to sense something. We call that the analyte. And in order to sense that selectively, you need to have a recognition domain. So we work in that space. Often what we're trying to sense is a biological molecule. So our recognition domain are often proteins. So we do a lot of protein prototyping. And here, let me go to a laser pointer here. Be a little easier to show. So we've done a lot of work in cell-free protein synthesis. So being able to rapidly go from an idea of a protein and producing it in our lab and testing it. That needs to link. So once it binds, you have to transduce that information. So we use two different transducers. One is a radio frequency-based platform, and I will highlight two papers from this space. And then we also work with these near-infrared probes, and I'll tell you why specifically those two frequency domains. But this will be the other paper we'll highlight. We've also done some other crazy stuff. So I had a talented biochemistry student where we got interested in spores, these crazy organisms that can go dormant for millions of years and wake up. So thinking about engineered spore-based devices. Sensors aren't useful unless you can actually read the information. So we do a lot of work within these papers as well as standalones, building low-cost readers so we can deploy them in the field. And really what makes our kind of the problems that get us really interested are these things where you can't have a wired system between your reader and transducer. And of course, here top of mind today is a plant system. So if you're in a field, you could wire everything up, or you could get wireless modules, but each of those come at a cost. So having truly passive, low-cost battery-less systems is of interest. Looking at bioreactors is another closed system we're keen on, trying to monitor process conditions without having to have wires go in and break sterile conditions. And then of course the living systems like our body, having sensors that can transduce information. So really, if you take away this constraint of being able to wire, there's what we use is electromagnetic radiation, so beaming information in and out. And if you look at you got some doodle notes here, sorry. So if you look at, in this case, this is electromagnetic radiation and absorption in water, which is a common material that we have to go through either in soil or tissue. There are a few bands that operate really well. So gamma and X-ray, you go to the doctor, you break an arm, it gets scanned. Those really high energy waves can penetrate through your skin, no problem, bounce off, and you can get information in and out. There's also an interesting band in the near infrared where there's very low absorption. And so that's where we tune our near infrared probes, which I'll talk about. And then we also play here in the very low energy system, which also can go through water and plastics and soil. And these are the shortwave radio frequency sensors. And I'll talk a little bit about those too. So in order to cover the platforms, let's talk about the near infrared probes first. This comes off of my graduate studies where really it's an interesting class of materials. Anything engineered in that nanoscale have these really interesting optical and electronic properties. And in the case we use single walled carbon nanotubes. So they can be excited. So you shine a laser or a high energy source at them. They excite, and then when that exciton recombines, they fluoresce. And based on the size of the nanotube, you can have different band gaps and different colors or different frequencies at which they fluoresce. And those result in these different colors. So each of these are different diameters of nanotubes. And the way we use them as sensors or as probes is their surface is really sensitive. So if something binds to it, it will change the fluorescence. And our job is figuring out how we can engineer the surface to be selective to what we're trying to bind. So I'm going to just cover my PhD in a single claymation here. So my PhD was looking at protein binding, and then it was specifically focused on antibodies and looking at the sugars on antibodies, so glycosylation. So let me see, I have to get off my pointer here so I can actually point. All right, here we go. So what we do is we wrap the nanotube, in this case, with lectins. So they're selective to sugars. And when the right sugar-lectin combination would occur, it causes a modulation of fluorescence. And then if it's non-selective, what we're trying to do is passivate the surface. So only the specific binding is what you see. So there it is, whole PhD in one claymation. So voila. All right. So one thing that we've done in my group, and I'll talk about this, is rather than looking at binding, we're really interested in looking at things that degrade materials. And that's where soil gets really interesting, because there's a whole complex host of organisms and enzymes that can break down biomass. And so that's just a teaser of what I'll talk about. Now with the other platform, with the shortwave radio frequency, this is a side view of that coil. And what we do is we excite that coil, again, with a specific frequency. And what it does is it's able to absorb that energy and resonate. And so it's an inductor and capacitor, and this energy just kind of bounces back and forth at a set frequency. And what we do is we monitor, so we ping it with a bunch of frequencies and we determine what frequency it's resonating at. And in order to make a sensor out of it, we change the surface. So if something binds, much like the nanotube, if something interferes or comes in contact with the surface or changes that permittivity, it will shift the resonant frequency. And so it's a very handy, kind of clear, simple readout. And then again, what we're trying to do is just change what we're putting on here. And so I'll talk about some enzyme measurement that we've done with this, with soil, with this platform, as well as a very obvious choice, which is if you put water in contact with this resonant coil, it will shift the frequency dramatically, because water and air or dry soil have a very different permittivity. And so this is really easy to render into a very low cost soil moisture sensor. All right, so here it is highlighting. So we're going to go back in time to when I was at DuPont, and I'll show my first forays into soil, and that will be a very short vignette. And then we'll talk about the first version of the resonance sensor, and this was focused on enzymes. Then I'll talk about enzymes being measured by these near-infrared probes. Then we'll talk about more recent work looking at the soil moisture. And then if time allows, we'll look at this nutrient sensor and enzyme mining, which is work that we're currently doing. All right, so this is back in time. So the very first time when I was working at DuPont, where I got kind of interested in soil questions, was working at their field sites. So one was these in-soil rhizotrons, so built these little robots that, well, not really a robot, imager that could go down and image root systems, coming up with automated systems for measuring cyst nematodes, looked at a lot of color analysis, so walking through the field, kind of helping get more data that an agronomist could use. And then this was a really interesting project where we were looking at on-demand fertilizer release. And so what we did is built a little buggy that could drive over these sachets of chemicals that would be buried, and it would heat them up and essentially pop them. And so you could get this kind of on-demand release of material. So very first kind of touching systems of soil and getting really curious about what's there. So then I came here to Iowa State and was happy to find a like-minded individual, Dr. McDaniel, who is also as interested about soil. And we had this project where we had this resonant coil. And the thought was, well, there's lots of enzymes in soil. Could we come up with a system that could be buried, that could measure maybe over time or just at a short time vignette, different activities? And so this project was born, and I'm just going to walk you through it a bit. So one thing we did is we created these coils, and we tried again to make the fabrication really low cost. So how we fabricate these is we design the coil, and then we print it actually just using a craft cutter. We've gotten a little more high tech since then, but you come in and you get what you can and go fast. So we would pattern these onto copper that's coated on a polymer, and then that indelible marker or Sharpie would act as a mask. You would etch away the material that you don't need, and then this coil could be excited by this vector network analyzer. And this is a system that you can buy, but it's one that also we've done a lot of work to make really low cost. And all it does is, it just sweeps frequencies, so you can ping it, and then you can figure out what the resonant frequency is. So with that working, we came up with this idea of, well, let's put a substrate on the sensor. In this case, we're looking at proteases, so you can put a substrate of different hydrogels like gelatin, and we would coat it, and we would create a small well in the middle, and we'd add enzyme, and then over time that enzyme would degrade the sidewalls of that gelatin, and eventually the gelatin would degrade to the point where now it's just all liquefied. And just proof of concept, right? I like these kind of quick tests to make sure it's something that we can work on. We looked at the shift, right? So going from a gelatin plus the enzyme going to complete digestion, there was a very clear shift that we could track. So now it gets really interesting, and this is where you have to get a bit more technical, right? So we wanted to flex our engineering muscles a bit, and so we wanted to model the transport. And this is a really interesting problem because your boundary conditions are changing over time. So this is just a movie of, in this case, it's enzymes put in with some fluorescent pink beads. So that way you can monitor it over time. And so as the enzymes are degrading, right, it radially increases that surface area. And so you have this dilution of the enzyme occurring simultaneously to your boundary condition getting bigger and bigger over time. And so just using a standard equation for a reaction diffusion equation, we set up these boundary conditions, and this was the very first time I played with one where it's moving, so that's called a Stefan boundary condition. And both, we just came up with an analytical, it's not analytical, a numerical solving solution to this. And so what that allowed us to do is say, okay, now what we can do is we can model this, this change in radius versus time, and we can relate that change in radius to frequency. And so now we can get the frequency rate as it changes, and we can correlate that to enzyme, and then correlate that to the turnover number of the enzyme. And so this was all modeled, and then what we did with our experimental data is did the same thing where we could run them at different enzyme concentrations, and then map it to our model and get kinetic fits. And what was cool is we could also play with the gel itself. So you could put in materials like PEG or different plasticizers to slow down the degradation. So if you wanted a sensor that would degrade slower, or if you wanted something that to degrade quicker, you could do that. So then the soil comes in, right? So now what the main goal was trying to make a little device that could sit and get exposed. So this was the rudimentary device. It just had a coil, had a little access port so that water could leach in from a wet soil. And if there were enzymes present, it would start degrading. And sure enough, if we had soil, I think this was from one of the experimental fields that Marston frequents at. And if we just put it on straight on, we could get degradation. If we autoclave that, we kill that signal. And then if we add extra enzyme to it, just spike it, so it's like a super soil, we definitely get degradation. So kind of an interesting touch point, but one that would have a little bit of practical utility problems. The main one before this study, or what we knew would be a problem is here in this bottom reader is our antenna to pick up the sensor coil. It's very sensitive to how this coil is placed. So this experiment, this is where these are like the disclosures, right, to papers. So here's the disclosure. This works great if nothing moves. So if this coil sits on it for the experiment, no problem. You can see it, not an issue. But if you come and like in an actual field usage where this puck would be buried and you'd come back and measure it, that would be an issue because you'd have a different frequency. Not a huge difference, but enough that it would swamp out the signal change. And so we went back to the drawing board, and you'll see one of our later ones where we've tackled this issue of position sensitivity, okay? But we'll leave this one here. And now we'll go over to the near infrared work. So same thing, since we had been working with Marshall, we decided to do it again. So here he is again on another paper. And we wanted now to take our smaller probes, our near infrared ones, which again can measure enzyme activity, but look to see if we could go do that in the field. And so again, the motivation is this, large area of precision agriculture. I'm preaching the choir, right? There's lots of heterogeneity. So rather than sampling one, let's sample in lots of spots. And so how we build these sensors, again, that carbon nanotube is a very sensitive probe. The surface, any change to permittivity or dielectric around it will change the fluorescence. And so to render it into an enzyme-based sensor, we take whatever substrate that the enzyme is going to chew on. So a protein for a protease, cellulose for cellulase, starch for an amylase. And then what we do is we place that in solution with the nanotube and we sonicate it. So we disperse the nanotube, and some of this material will bind to the nanotube and keep it in a soluble form. And so then we can centrifuge, pellet out all the things that didn't suspend, and then that supernatant has our solubilized sensor. And now we can take that, and it's really handy to use in a well plate format or cuvette, because you can just take your sample of interest and add it to it. And the other thing that's really handy, so one plus of it is any substrate can be placed on there. So if you have an enzyme that's working on a very exotic substrate, or you have some proxy probe that you're using that's not really a good indicator of the actual activity, this is really useful because you can actually test the enzyme on the substrate of interest. The other utility is because it fluoresces in the near infrared, you can put these in really dirty samples. We've done manure, we've done soil, whole cell lysate, because it's not in that visible region and there's not a lot of background absorption or noise, you get a very clear signal from it. And so what we do, again, simple experiments, is we first just wrapped it in a carboxymethyl cellulose. We put cellulase in there and let it sit overnight. And sure enough, our mental picture of these things degrading and getting hydrophobic and quenching and crashing out of solution, it actually occurred. So they all fell out. So we don't want to sit overnight for our assay. So we built a little low-cost fluorimeter that uses an LED and a electrically amplified detector. And we would, again, wrap it in a modified cellulose and add cellulase, and we'd get this very nice degradation pattern of the nanotube. And so in the paper, the very first paper, this one down below in 2018, we explored lots of different enzymes and showed how modular it was. But then in this follow-up work, we decided to look specifically at soil. So we had soil, and we looked at proteins and different types of oligosaccharides. And what we did is, again, we wanted to take it out in the field. And so we had this plot. We looked at a couple different sites as well as different areas within the sites. And we could correlate it decently well with the assays that were then done in lab. So the kind of existing assays. So these ones were done with Marshall's student, and then these ones were done with the nanosensor. The other fun part was actually going out and taking tests in the field. So rather than having to put it in a cold box and bring it back to the lab, we built this very rudimentary device. This has gotten a lot better. Yeah, as a disclosure, so Nathaniel, this guy, is now running this as a startup. And so his boxes look a lot nicer and are more robust. This one, you drop it, everything would break. So, but what we did is we made it work such that you could do these in-field protease activities and just kind of got little simple heat maps of if we sampled, down a row and measured the protease activity, you could get relative activity amounts. And he's done subsequent work, again, looking at kind of blends. So if you take those different chiralities of nanotubes and wrap them in different materials, you could have one that's geared towards biomass, so you can measure at the same time lignin and cellulose and hemicellulose degradation. Also kind of teasing out endo versus exo activity. And again, this is all just kind of ongoing work as we keep exploring the limits of the sensor. So yeah, so that's where that one is, and is very applicable to measuring soil enzymes. All right, so now we're going to go back to we're moving forward in time. So we've thought a lot about this problem of positional sensitivity. And I had two very clever students who came up with this idea of how you could overcome that issue. And they did that in this paper, but then they also applied it to moisture sensing. So again, water has a very different permittivity, so that's a pretty easy thing. You really don't have to change this coil in any manner. And I'll explain the geometry here in a second, but what it is this inductively coupled extender. So we called it an ICE, all right? Now, the reason we got into fluid measurement as well is we've also had other studies looking at moisture. So this was another one where we had an issue with positional sensitivity. So Adam, one of the students here on this paper, he got interested in measuring moisture from a wearables point of view. So a funded project with firefighters looking at under PPE, so under the personal protective equipment. They build up a lot of moisture, and that's one of the big issues of heat stress is oversweating. And so we created this small puck that could basically wick in the sweat and then measure the amount of sweat that's been collected under the coat. So you could just measure through the coat. But again, the issue was you'd have to align this antenna pretty carefully to figure out what the measurement was. So that led us to this innovation here. And it's, again, very simple. That's where innovations are the best, right? Because they could be replicated easily. So before we were measuring a coil with one antenna, and there's a lot of positional sensitivity. This mutual inductance between these two is very sensitive on position. So my students had this idea, well, what if we put another coil between it? So now this is the coil you're reading with, and it mutually couples to this side. And now that excites this kind of dog bone looking thing, okay? So now this is excited, and then these two are also mutually coupled. So you have this antenna that's coupled to your sensor. And now what we do is we just create a sensor package where this coil is affixed to this one. So the position of that is fixed. And now when these two vary, it doesn't matter, because if the peaks, so if the resonant peak of this is different from this one will shift all over the place depending on position, but these two will stay consistent. And so very simple, and it's like, hey, that's great, build it, go try it, test it. And so that's what they did. Well, they were smart. First, they did some modeling of it. So this is a model where in the case of moving these two coils around, sorry, let me get back my pointer so you can see a little bit better. So moving these two around, you get a whole bunch of noise in that peak, which is that we'll call it the kind of the reading portion of the extender. But the sensor part would stay consistent. But now if you change the permittivity around that sensor and change position, you get a very nice clear shift in your sensor peak and not the noise that we typically had. And so we then decided to build it. And again, so this sharp peak here is that reading coil, and then the smaller peak here, because there's loss of energy through the system, but you can still see the peak, would be with the sensor. And so now how this looks is we have a long extender. They're just two wires that go down to that coil at the bottom. And those can be as long as we want, but we went as far as like a meter and a half, and we can then bury this part, the sensor part, down in the soil. And then what we can do is we can look at how that shifts. So this is shifting up this way as the water content changes at the soil height. And now you can also, the way this was translated out to the field was a stake. So just kind of a smart stake, if you will, that you could pound in at the end of a row that would have this coil portion at the end. And then an agronomist or a rover could come by and scan that top part of the stake. and get a measurement of the soil moisture. And so we did that in our lab as well, having different heights here, trying to look at consistent gain. So that's the change of your sensor signal versus moisture content. Also really important to track that with existing tools. So this is a veneer soil moisture sensor, and we could track pretty consistently. I think R² is 0.9-ish or so between the two. But like many other sensors, things that would plague it are cracking in the soil. So if you got like, big chasms and you get a lot of moisture right by your sensor, that might be different than if the soil was more homogeneous. So like in any real system, you might have to measure it and get a calibration curve in your system. But yeah, the main exciting thing was that positional sensitivity was now gone. So the sensor could be left and it could be read over time. So over that growing season, my student could come back and just measure it and then correlate to changing weather and patterns, moisture, as well as moisture at different depths, because you can have stakes of different depths. This is one example of that from this paper. So we had soil moisture. So we built a column of soil having 3 different orthogonal sensors, and we would dump the water in at the top. And then as that moisture is coming through, it's very insensitive until it hits this very bottom sensor. So when that Vermeer sensor goes off at that depth, our LC sensor clicks off instantaneously with it too. And so you could also profile different depths, especially if you had different frequencies on the same pole. All right, can be done on smaller vessels too. So this is looking at positional reproducibility. So being able to come back and scan this multiple times was important. So then that way you could still build up trends or some calibration. All right, so that's what we're doing with soil moisture. This has naturally led into stuff in the soil moisture. So we're really interested in looking at ions, and this has been something that's been ongoing for a long time, and it's harder because we want to be selective. So now we want to put something on the sensor, near the sensor, to promote a signal from one ion of interest. And of course, it's no surprise what's the ion of interest for looking at nitrates. And so what we do is we look at different ion exchange resins, which we've again been collaborating with Marshall on, looking at their selectivity to pull out or to absorb ions that we're not interested in. And so that way we can selectively transduce ions. Coming along decently well. Probably where it'd be useful is kind of at the end of larger runoff areas, so like at the end of tile drains. And again, these sensors aren't smart in the sense of sending off the signal. So if you want to get data from it, you have to come by and scan it. However, the cost of them are such that they're very low cost, so you could deploy them in many different spaces. So that's where it's useful, is if you are kind of already walking a field or have a rover or a drone or whatever, they could quickly scan them. But they could be left there for a long period of time, and you could have many different sites. All right. The other thing from soil that I wanted to highlight today, and then I'm happy to take questions, is this idea of learning from soil. So one of the things that I've been really interested is just how wonderful the process of all these unit operations happening in soil are at doing things that we try and recapitulate outside the soil and we're not very good at. And one of those things is degradation of products. So biomass I've already talked about. Right? If we try and create a complex system of enzymes to break down biomass, it's a very crude approximation, us trying to do it with enzymes and trying to achieve the efficiency of which soil can do it. So there's lots of learnings in that space. Another one that I've been active in is looking at non-biological material degradation. So I'm sure you're aware of interesting soil-based microbes and enzymes being sourced that degrade plastics. So we're collaborating with a couple teams that have done genomic mining and just kind of scraping of different samples. And from that, they prospect a large number of enzymes. And then what we do as a team is we try and model those, their structure and selectivity, to then down select ones that we want to test. And so that uses that cell-free protein synthesis, so we can create an enzyme, and then we can screen it with our near-infrared probe. And then that allows us to figure out from that test which ones are of interest, so that way we can feed it back into the model and then rapidly iterate to try and find enzymes that can solve large pressing world problems like waste management. And yeah, here's just another highlight image of that process. So we have our own gene synthesizer, so we can create a bunch of different constructs. We have these little minimal templates, create them into enzymes, and then we can add our probes and test their ability to degrade those polymers. So in this case, it's not a biological polymer, but a synthetic one. So, oh, and then this is showing us some process. So again, this is all very kind of new stuff, but this is a polyurethane that's attached to the nanotube, and this is one of those prospected lipases that's a hit. So it actually can degrade the material off that nanotube. So yeah, exciting. Okay, the other thing is every time I give a talk, I like to get on a soapbox. So here's my soapbox, take it for what it is. But I am a big proponent of us doing research and having it do something. So coming from industry, I guess I have a chip on my shoulder that we owe it to society to solve some problems, and we're training next generation of scientists, and we should always keep this top of mind. And so, as was mentioned in the intro, it's kind of a hobby of mine to go and create startups. I don't golf; that's what I do on my spare time. And so, if there are individuals within your program that are interested, I'm happy to help with that. ISU has a lot of programs to help cultivate and help students and scientists move innovations out and translate it, but there are also a lot of great programs. I'm biased because the NSF ones have been really supportive for us, but there's customer discovery programs that give student budget to go and talk to people who might have the problems and figure out if their technology or management practice or innovation can be useful. And then there are other funds that can support your lab to do that translational research. And I'm a huge fan of this PFI program. So if you're interested, I'm happy to talk about that. So with that, love to take questions, but again, want to highlight all the great students who did this work. I already gave a shout out to our wonderful collaborators. Marshall, you're not on this list. Okay, I got to update this. Marshall is one of our collaborators here at ISU. Yeah, happy to take any questions. Yes.
For the soil moisture device,
Yeah.
it's like a TDR that you build a bit and then you have to install different ones.
No, so what we're trying to do is create.
Can you repeat the question?
Oh yeah, sorry. So the question was, is it like a TDR where you'd have to like dig a channel and stick them in on the side? The one that we deployed, we were trying to create something that would be really easy to insert. So a stake that could be pounded in. And so the idea would be that that coil would now be on the external parts of that stake. Ended up, we're not very good at making stakes that are robust enough to pound into Iowa soil. So they're pretty easy to kind of rip. And our biggest issue is if the lining would get torn, moisture would start filling kind of that center pole. So some basic engineering could fix that. But yeah, the goal is to try and create it so you don't have to dig a hole. We ended up having to do that on a lot of them.
And then this has like a data.
Great question. Yeah, so that goes back to the cost. So I think we're all very familiar with either data logging systems or ones that have like a Zigbee or something that can beam it off, you know, beam it to a cellular tower if there is one. What we're trying to do is get rid of that system, so that way each of the nodes would be like $1 or less than $1. But they're dumb. They don't have any data logging capability. So the only way to get data from it is to go and walk by it with that reader, either walk by or drive by. And so again, that's that trade-off of you don't get continuous data in the sense of what a data logger gives, but you can have tons of them. You could have, you know, in a field, you could have thousands of them. You just have to walk the track or drive over it.
But each one is, when you use one that can measure different depth, or you need one per depth.
Great question. So in the one that we deployed, there were one per depth. but it would be easy to have it such that you'd have those coils. So think of a rod that would have coils at different positions that would then be at different frequencies. So you would sweep it and you'd have like 3 peaks, and those would be your 3 different depths.
When you calibrate, how accurate is the measurement?
Yeah, so that was that one where again, this is more of a controlled system, right? But these were these tests here where we would have our orthogonal moisture content sensor measuring it as we were measuring it with RF frequency. And so we do this multiple times, and we could get parity again within that kind of over 90% certainty. But it's a funny thing in any form of sensing. It's now you start pointing fingers of like, if there's variability, is it you? Is it me? Is it you? So yeah, it's good to test against what others are using.
We have a question online, a couple of them. I'll start with one from Dr. Hornbuckle. He said, "I'm happy you are here at ISU soon."
Thanks, Dr. Hornbuckle. I'm happy to.
Instead of being in industry, we're glad you're here, for sure. What is your sensing volume of your soil moisture sensors? Please give dimensions in centimeters. Okay, great. Yeah, so they can get as big as you want. This one that's in this picture right here, sorry to repeat. So we're looking at what's the volume or area that can be sensed. So in this mode, this one was about 3 centimeters across. And the penetration depth it's looking is about 1 to 2 centimeters. So think of it as kind of a half, half sphere that it's measuring. But that depth can be tuned. So in that biomanufacturing work that I talked about, where we're monitoring cells and different forms of bioreactors, sometimes we want to look at bulk. So we design the sensor such that its electric field goes deep versus ones where the cells are adherent. We just want to look at a very localized field. So that can all be tuned based on the geometry of your sensor. Next question was from Dr. Horton. He said, Adam Carr took my soil physics class, and he said that he was working on a soil water potential sensor. Did he have any success with the sensor? Yeah, okay. First of all, thank you for the training to Adam. That was super useful for, so anyway, yeah, comment. My student took a soil class here. I don't know. So there was a lot of work done, especially in collaboration with Corteva, even in that form, the ion sensing, which I'm not at liberty to share just because it's tied up with Corteva. But we are doing a follow-up study of the ions. Now with the water, I am not certain, so I'd have to go back and check. Really interesting. This is from Dr. Thompson. Thanks for all the great work that is relevant to soil processes. To detect enzyme activity in soils with the sensor, is it necessary for the enzyme to be soluble in water, or could it be bound to clay and its activity could be sensed? Or could you measure how activity of the enzyme is reduced by the direct association with a solid phase? Great. Okay, I'm not going to repeat that question because it was a very good and long one. But if you're online, you can read it, I presume. But yes, so the idea is, can we tease out this difference between enzymes that have been secreted and kind of free floating versus ones that are tied up on clay or in otherwise immobilized? The answer is we can do both, because actually in both cases, water is still involved. So we call it a hydrolase because there's hydro water involved. So that in order to have the catalytic activity occur, water needs to be somewhere present in the system. But you're completely right that sometimes it's a free floating enzyme that has a lot of mobility versus one that's tied up. Now we have subsequently done stuff with like farm feed enzymes. So with those, they're immobilized on pellets. You still can measure activity, and it's definitely lower than ones that are free floating. So that would be a very interesting study, one that we would be happy to do. If you had samples of soil where it was more clay bound versus ones that are free, that would be something that we could screen. And again, to that point of being able to either do it out in the field or having the capability to run more in the lab. Because that's something that we found working in Marshall's group and subsequently is there are existing enzyme assays, but a lot of them are a pain. Yeah.
Yeah.
I'm curious how much thought you've given or much knowledge as far as that top layer of the soil will be an active machine layer.
Yeah.
To be tilled,
Yeah.
planted, and so on.
Yep.
Are you thinking that? You mentioned like the rod could be really cheap.
Yeah.
So disposable.
Yeah.
Or pound it down low enough so it's below how layer you left, not damaged by pounding?
Great question. Yeah. So right now we're cheating in the sense of they're all research farms where you go back and you grab things. So yeah, we would just do them at the end of rows of different trials that have moisture related things that they want to measure. We have given a little thought. So we did one paper where we looked at these sensors, what they can be printed on, what they can be made out of. They can be really made out of anything. This would be conductive enough to conduct electricity. So, we've done iron powders and different metals. That would just kind of get into your cost of goods and what you want to actually leave in your field. I think easiest modality right now would still be something where it's like end of row that you would want to just come pick up at the end of season. But conceivably you could have one that could be deeper. One issue, they can't go too deep though. So that's one problem is because these are unpowered, you're dealing with near field coupling. So if you are trying to get farther away, your coils get bigger. So you can manage that to a certain extent. But it's never going to be like meters away. We're probably talking tens of centimeters. So, yeah.
The passive, I'm not excited about that strategy.
Yeah.
But if it's 30 centimeters below the soil.
Yeah.
You still drive over it and get.
Yeah. So that goes to that coil size, and also the power of your system. So a lot of the things that I showed, these little, like this guy. Well, this is a little bit more expensive, but we have these like little portable readers. They're very low power, and so we're always getting in a few centimeters to couple with it to get a strong signal. You can come up with systems. We're working with a collaborator here at Iowa State to build our own pseudo VNA that's a little bit higher power and can read further distance. But again, it's never going to be something like where it's buried where Marshall's sitting and I'm trying to scan it. You're going to have to get close to it. But yeah, it could. The nice thing is it's in the frequency range where as long as you're able to mutually couple, you can still penetrate through the soil to see it. Other questions? Marshall.
Sorry, John, did you have a question? You look like you're about to raise your hand. Go for it. I get to talk to Nigel all the time. Go for it.
Okay, so you know where I'm coming from with this. So I'm thinking of fine scale.
Yeah.
Could we use this type of technology to say measure concentrations of nutrients, enzymes, or whatever? Look at the gradients next to it.
Yes. Yeah. So okay, now you're pulling together. So I showed you my Rhizotron stuff way back when. One thing we're doing, and this is actually something that we've built into this project, is looking at spatial heterogeneities. So there's the reason this comes up in like plastic degradation is enzymes are active in different areas. So yeah, what we're doing is building imaging systems, right? So rather than just collecting all the light from a well, these nanosensors are nano size, so you can get spatial information too. So just like you said, if you had a root, that was laid out, as long as we have a imaging system that works in the near infrared, so it can't be a silicon detector, it have to be like a in-gas detector. And you're imaging that system, you could see, you can map spatial activity. We could readily do that with enzymes now. We are also building classes of sensors that look at smaller molecules, some metabolites, like ATP is one that we're building. And then also proteins. So if there are things that are being secreted, that would be of interest. But yeah, that's something that we're definitely keen on and trying to build up capacity to do. Questions?
Well, I wanted to ask you about the kind of startup businesses that some of your students have gone on to run, and if you would just talk a little bit about that. It's not something in our department we're familiar with. And,
yeah.
Are you still really involved, or give them their own space and let them find their way? Or
we just can't stop working with each other, so that's the problem. Really. So yeah, I really enjoy working with my students, so it's always fun to see where that relationship can continue. And so usually, what initiates it is a student who has that interest, right? That kind of entrepreneurial bug. And it can actually just start with like, hey, I'm working on something that I think could do something, right? And you just work in that space. And most of my students have done this I-Corps program. Either there's a local I-Corps program, there's also a national I-Corps program. And what that does is it allows them to sort of put their technology to the side for a bit and talk to people who are in the space and actually figure out what the problems are. So the student, Nathaniel, who's running Enzymebased, he has done I-Corps a couple times, and what that allows him to do is talk to different industries that use enzymes to figure out where the pain points are. And where I come in as a PI is, supporting them in the way that I do best, which is getting money. So I try and time it so we can get some of these grants to work internally, but then I try and time it based on their graduation to get some of these small business innovation grants so that those hit by the time they graduate. So we have a nice runway. And that's worked in a couple cases where the funding comes in and then it's sort of them building their own super postdoc, right? They can be their own PI at that point. It's a lot of learning, right? Because now they have a team that reports to them. And usually, to answer your point, usually it's their chief on the technical side and operations day to day. And then I come in kind of more on the business customer interactions, sort of interim president at the start. And then as it grows, I try and excuse myself as quickly as possible because I'm really good at getting it off the ground and flying, but then, when it's doing the transatlantic flight, that's when I don't want to be on the plane anymore. So yeah, but it's exciting. I mean, it's a great, and I think where the passion for it came is coming from DuPont. A lot of these larger companies are looking to us for innovation, right? So that in my bio, I mentioned I went from doing research in DuPont to DuPont closing central research and saying, we just got to go find innovation elsewhere. It's too expensive to justify to our shareholders. And that's happening in a lot of companies. And so they look to academia and very intelligent and smart students who go out and say, hey, I've got this idea. And so, I see this synergy of where we can fit that. And yeah, it works, right? If we can find the right problems and find the right partners, we can solve problems. Any other questions? Okay.
Follow up on that. So you mentioned being a short time president or something.
Yeah.
I think for a lot of academics, it seems like really defined to manage.
Yeah. So yeah, rest assured, we have a great conflict of interest panel here at Iowa State University, and they're not scary. They're not there to beat you up. They're there to manage the issue that could occur. And so whenever you kind of step over that line of having some vested interest, so you've incorporated, there's a company, you proclaim it on your conflict of interest statement, and that's students and faculty. And then what you do is you set up a panel and you talk about it. You say, okay, here's my research line, here's what the company does. If there's crosstalk, this is how we're going to manage it. Usually it's quite simple. It's, you have a strict firewall. So in the case of all these startups, they're not in Sweeney, they're not in our building. They're off at different incubators north of campus and also in the research park. So there's, a clear separation of church and state. So there's no usage of money or resources. And then the other conflict is usually personnel. So if there's a student involved, there's management plan put in place. But I usually try and time it so that students graduated, so that conflict's not there. What else? Yeah, and then you get to put a disclaimer slide, legalese stuff on a lot of things, just to show that you have other things cooking. But yeah, it's not too bad. I mean, usually the main thing is just time management, right? You just have to be really diligent figuring out how you do this. So how I manage it is my early mornings, I'm always down at Research Park. So I see the employees at the start of their day, and then I come up here and do my ICU duties. Any other questions? Okay, sorry. Yeah.
No, that's good.
Appreciate you all coming out.
Let's thank